Hi everyone, this is Dr. Santosh. Today I'll be talking about the structure of DNA and the history behind the discovery of structure of DNA. Structure wise, it is a right-handed double helix, and this structure was discovered and proposed by Watson and Crick in 1953. So as you see, you can see the structure of DNA. The structure of DNA at its backbone, if you see the structure, analyze the structure of DNA. At its backbone is made up of phosphodiester bond and the sugar. And at the center here, everywhere, this center portion, that is, if you consider it as a stair, the ladder, the steps of this stair, it is made up of bases. And bases are there, the two bases, one will be purine, another will be pyrimidine, always, and those will be in hydrogen bonding, and that hydrogen bond help that DNA to stabilize that structure. And not only hydrogen bonding between this, one is hydrogen bonding between these bases. Other thing is there will be the pi stacking or base stacking between the successive base pairs. So that will also help stabilize this double helix. Okay, so let's see the shape of, like, size of this double helix. So if you see the size of this DNA, the DNA double helix is a 2 nanometer molecule in its width, and when you see lengthwise, one piece of this DNA, one helix, one turn of this DNA is 3.4 nanometer, and distance between two base is 0.34. That means one helix contain about 10 bases. More precisely, 10.5 bases is present per helix in balanced or bDNA. So this is the DNA that was proposed by this Watson and Crick in 1953. So what the main thing we need to know is DNA is double helix, it is right-handed, and the breadth of this DNA is 2 nanometer, and then pitch is 3.4 nanometer. And what are the bonds that stabilize this double-stranded helical structure? Actually, single stranded of this DNA is made up of this phosphodiester bond and sugar as backbone, and then bases are present at the center, forming hydrogen bond and base pair stacking. That gives the stability to DNA as double helical structure. So discovery of this double-stranded helical is quite interesting. So this bDNA, balanced DNA model was given by these two scientists, James Watson and this Crick. So Watson and Crick, they proposed the model of double-stranded helical model in 1950. It is very interesting that these two scientists, they did not do any research work. Rather, this discovery of DNA is a conclusion from their part. Actually, they utilized the research done by many scientists from 1860s till 1953. Discovery where most important research work were done by Rosalind Franklin. She was studying DNA by X-ray crystallography, and she has done so much of work. Ultimately, she got cancer and she died she got ovarian cancer because of that X-rays, and she died in 1958 because of that cancer. And her contribution is very big. She has made a very big contribution for discovery of double-stranded DNA. And what it used to happen is she used to capture the X-ray crystallographical images of DNA, and Wilkins, without her notice, he used to keep data to Watson and Crick, and they used to figure out the structure of DNA. They used to read the data, and they used to think over that the formation of DNA. So what they did is, in the beginning, they did not put this bases in the center. It is quite interesting to note that these two scientists, James Watson and Crick, they, while discovering the double-stranded helical model of DNA, they did many mistakes in the beginning. What they did, the bases that were there inside, we studied on now, they put those all bases outside the double helical structure. And now they showed that structure to Rosalind Franklin. If that structure was right or not, then she dismissed that structure. That could not be right. And then there was a scientist called Linus Pauling. He was an eminent scientist at that time. He discovered the actually alpha helical model of this protein. So he proposed a triple helical model of DNA. So they were a little confused, but the breakthrough in the discovery is the photo captured by Rosalind Franklin. This photo is very famous with the name of photo 51. So when they got this photo through Wilkins, then they quickly understood that this is a double helical model, because the cross they see here, the cross pattern of X-ray, they quickly found out this is a double helical structure, double helical strand of DNA structure, because they knew the chemical structure of those bases. When they placed these two bases together, they found that there is a possibility of hydrogen bond formation between the bases. Okay, once they found that, it was a eureka moment for them, and then what they did, they found out the double stranded helical structure of DNA by placing base inside. Now, after he made a model and showed to Rosalind Franklin in 1953, they were very, Rosalind Franklin was, she gave her agreement. Yes, this could be the structure of DNA. This is the structure of DNA. The whatever the work she has done, it matches with that. Now, for that base pairing, they also got information from the work of this Erwin Charbagh. Erwin Charbagh in 1952, he explained to them about his finding guanine is equal to cytosine in all species of DNA, adenine is equal to thymine. So this helped them to find out this thymine should base pair with the adenine, and guanine should base pair with the thymine. So his work also helped him in discovery of double-stranded DNA structure. So after they discovered double-stranded DNA structure, now what happened is in 1962 they got Nobel Prize. In Nobel Prize, this James Watson, Crick, and Wilkins were added in Nobel Prize, but the Chargaff was not mentioned there. Okay, but we have to understand Chargaff has also contributed to the DNA structure, because his groundbreaking finding is purine is equal to pyrimidine and thymine is equal to adenine, cytosine is equal to guanine, was very helpful for them to find out the structure of DNA. Even though Chargaff wrote some letters to Nobel Committee, but he was not given credit for discovery of double-stranded DNA. Actually, the discovery of double-stranded DNA is not just their work; it is the work from 1960s. Many scientists has done. There are scientists who found out that DNA is present inside the nucleus. There are some scientists who found the individual nucleotide DNA is made up of a repeat of nucleotide. They found out the chemical structure of nucleotides. So the Nobel Prize, we should consider that it is a representation of contribution of many scientists. So why the people were in rush to discovery in double-stranded helical model was because that time there was many questions about how DNA is multiplied, how DNA, because people knew that DNA is a genetic, how DNA is duplicated, how DNA is transferred into other progeny, in upcoming progeny. Not only that, how transcription happens. Those kind of questions, there were many questions that time in the mind of scientific community. So those questions will be solved only when structure of DNA is discovered. So that is why the structure of DNA is a very groundbreaking discovery in the history of this genetics and molecular biology, and that opened the Pandora box for all the discoveries, many discoveries. Now we are in very advanced stages of this molecular biology because of the discovery of the double helical structure of DNA. So remember, when you understand the significance of this historical perspective, you understand the molecule better, and then you understand the structure, why you understand the fact that why we need to understand the structure of molecule. So understanding of a structure of molecule is the first step to understand the whole molecular biology. So this is why the understanding of DNA structure is very, very important. So please focus on the structure of DNA. We have done many videos on that, so go through those videos and understand the structure of DNA really well. That will help you understand genetics really well. Thank you.